welcome back. How's the rest of the semester going? I gotta give you like an early lecture. Anyone teach you anything interesting yet? Take that as a okay, so I think I'm teaching this one and the next one. Are these the last two lectures of the semester? Yeah. Okay, so hopefully they won't be too bad. I'm sure they won't be too bad. So this lecture I'm going to talk to you about is physical foundations for connectomics. The lecture I gave a couple weeks ago at the annual connectomics conference. But before I get into the details, I figured I should give a little background. So someone's talking to you about neurons already, right? And did anyone teach you that neural networks come up at all? Yeah. What did they tell you? Anything? Just yell it out. Neural networks exist. Anything else? That was a very short lecture. Anything else? You guys know about synapses? You know about, say, MRI, estimating connectome? Does the word connectome come up at all? No. Okay. Okay, so let me take a step back there and tell you guys what I'm going to talk about before we get into the slides. I can give you some examples. Okay, so what I'm going to be talking to you guys about today is a thing called connectome. So the brain is made up of a bunch of stuff. People have probably gone over basic anatomy of the brain. Okay, so what's in the brain? What do you got in there? Cells? You know when they discovered that? Brains were made up of cells? I want to teach you the history of this. You know when cells were discovered? When? Does anyone know? When did the first person think there was a cell? Does anyone know how someone figured out that there were cells? Yeah?
They put a cork under a microscope.
Yeah? That's right, they put a cork under a microscope and they saw cells. When did they do that? 1665. That's the answer to that question. Robert Hooker built a microscope and he worked under a cell, and he saw there's a bunch of cells. And then people started thinking, oh, maybe stuff is made up of cells. And like 200 years later, people said organisms like plants and stuff, seems like they're all made up of cells. It's just kind of a mind-blowing thing. It was like your great-great-grandparents didn't think organisms were made of cells. Just thought, whatever. I don't know what they thought. It was weird. But eventually people had this idea called the cell theory, which all things are made up of cells, all organisms. And then it's a question about, well, the brain is inside organisms, so the brain must be made up of cells. So, like, let's say around 1850, what do you guys think people thought brains were? You're living in 1850, it's like 107 years ago, and you know you're a doctor, and someone gets half their head blown out or whatever. You look in there, what do you think it's made of? Cells? By cells, right? Yes. And then you think they're connected? They're just all like red blood cells just kind of floating around there, not connected? That was one idea, just a bunch of cells. Were there any other ideas at the time? How many cells were in there, did they think? 10? Yeah, I don't know what they thought. Some people thought there was one cell. One big ass cell with like billions of nuclei. It's called a syncytium. And it turns out they weren't all wrong. There's still some organisms with syncytial cells. It's kind of annoying dealing with them. But there are such organisms, and that's what people thought. There was one big cell and it had lots of nuclei. Or the other thing they thought is there's lots of cells and they're just not connected. And then how do they figure out that that is not exactly right? You know what happened next? I'll give you a hint. It was like in the late 1800s. It involved the microscope. It involved the stains. Has anyone told you about Golgi staining yet? So Golgi was a Mexican, maybe? Golgi Mexican. I think he was Mexican. Anyway, there was a Golgi dude, he had a stain, and then what happened? Someone looked on the microscope, what did they see? Are you guys just whispering synapse? Is that what's happening? Yeah. You just say it out loud. Okay. Okay, yeah, so Ramon Cajal saw synapses, and he said, holy shit, there's synapses. These is not one big cell. This is a whole bunch of cells, and they're connected to each other, and the brain is a network. Okay, that is like 100 years, 130 years ago, he had that realization. And then the next 100 years of neuroscience happened, and there's all this theory, all these models of how the brain works, from cognitive science, artificial intelligence. And the first people started talking about artificial intelligence as early as the 40s, modeling off of the brain, and the idea was the brain is a network. And then like 10 or 20 years later, you had neural networks. And then 70 years later, you have deep networks. All of that is based on this first realization from Cajal that brains are in fact networks of neurons. And other cells too, but for whatever reason, we always focus on neurons. Okay, so that was a really important realization. And everyone has kind of known that since the last 130 years. But in the history of neuroscience, what happened was it's really hard to measure what's connected to what. It's much easier, not just in neuroscience, in general, to just measure a thing. Right? Let's say you're in physics and we're measuring, say we're studying billiards. There's a bunch of balls. It's easy to measure one ball at a time. Say this ball is moving around, it has this volume, it has this mass, it has this acceleration. Once you introduce two balls, now there's interactions, and measuring interactions gets really hard. So there's two balls with only kind of two interactions. There's three balls, all pairs of them, there's more interactions. So there's four balls, you have all pairs and all three-way things. It quickly gets really out of control. You get lots and lots of interactions. And so measuring interactions between stuff is just fundamentally harder. It's not just in neuroscience, but just in anything. Measuring stuff is relatively easy, and measuring interactions is relatively hard. So from around the early 1900s till around 2000, like 100 years of people studying neuroscience, for the most part, what everybody did is they studied individual stuffs. Right? So they did it at different scales. So when you're looking at really little scales, subcellular scales, people studied channels, conductance, and maybe the nuclei, stuff like that. Scaling up to like what you can see with a microscope, they studied individual neurons. What are their morphologies? Where are they? What are their electrical properties? When you jab them and like shock them, how do they respond? And then you scale up to like macro level, doing like kind of human brain imaging kind of stuff. You can see where are the regions, and are there regions, and what if you electrocute someone here? What do they do? And it turns out they go like this sometimes, and sometimes they yell, and they do lots of weird stuff. And in the 50s, before there were laws, you could do that with people. You could, like, electrocute their brain and see what happens, and it's pretty cool. Now you can't really do that anymore. But anyway, the point is the whole 100 years of neuroscience, modern neuroscience, everyone more or less just focused on studying things on their own and not studying interactions. And it's not entirely true in some like model organisms, some invertebrate systems where they could study like 3 or 4 neurons together that form small circuits, like a central pattern generator. It's a kind of circuit. It's in everything. You turn it on and it does the thing. So that's how you walk. You just turn on walking and your leg just keeps going the same thing every time. It's how birds fly. It's how fish swim. It's how snakes slither, I guess. We all have these central pattern generators. So people study very small circuits like that. But to study big circuits, like, say, your entire brain network or all of the neurons in some animal, was really hard and mostly avoided, largely for experimental technology. And this is, even though people had been modeling abstractly networks of neurons for this whole time, there wasn't the data that people wanted to be able to fit these models. It was just conceptual, like, I think it should work like this. That was all of cognitive science. I think brains will look like this, but we don't measure because it's too hard. And around 2000, in the last decade really, that started changing. There's these new technological innovations that enable people to really measure lots of connections all at once. And they did this across lots of different scales, all the way from single neurons and parts of neurons all the way up to whole brains. And this field now of people studying connections of neurons is called connectomics. And it's a bastardization of the word connections. And you just add omics to stuff because adding omics makes it fancy and sexy. Right? So you don't just study genes anymore, you study genomics. We don't just study brain connections, we study connectomics. Right? But we study brain connections. So this last decade, people have been spending a huge amount of effort, experimental effort, generating these connectomes, trying to estimate all the way from really small organisms like a C. elegans, which just has 302 neurons, all the way up to humans that have, you guys know how many neurons are in a human brain?
90 billion.
How many?
90 billion.
90 billion?
Yes.
That's a pretty good guess. How'd you guess that?
I knew that.
How does that You know it because you read it, but how does that person find out?
That I don't know.
Does anyone know? This is such a great experiment. Are you guys guessing the answer? How would you figure it out? You want to know? It'd be really boring to count them all, right? Yeah, boring. So what would you do? There's a lot of them. How would you figure out how many? Yeah.
You just take a piece and like extrapolate, count how many were like small area.
Yeah, so you can really take a small piece of brain and count like 100 of them or so, then figure out how many, like what the volume of that is and what's the relative volume to the brain to multiply. People did that a lot. It turns out, let's say you do that in the cerebellum. You got one answer, and then let's say you do it in the cortex, and you get a different answer, and they're all by order of magnitude. And now you're not really sure what the right answer is. So that is exactly what people did for a long time. And they had a really high variance, and everyone got a very different answer. So now what do you do instead? This is such a great experiment. You guys can figure it out, I promise you. One way is counting them, right? What else can you count? Yeah?
Say representative samples from each region.
Yeah, so a different subsampling strategy, right? You grab some cerebellum and some cortex and some hippocampus. People did that too. Estimates turned out were still pretty variable depending on exactly which subsamples you did and what the volumes were and stuff like that. So what else could you count? What do all neurons have? Yeah.
Nuclei?
Nuclei. What do all nuclei have?
DNA.
DNA. And how much DNA is in a nucleus?
6 billion, 3 billion if you only have
What?
6 billion.
6 billion? 6.6 billion base pairs? How constant is that per neuron?
Pretty constant.
Pretty constant, right? Every single cell in your body has basically exactly 6.6 billion, up to an error bar that's more or less known. Okay, so now you know that. What are you going to do with that? Take a brain, and then what do you do? Put it in a blender. Right, obviously, it's the first thing you do in almost any experiment. Put a brain in a blender. Okay, and then extract the DNA. I mean, you can't extract it when it's like in fixed tissue. Extract the DNA. Weigh it, basically, how much DNA is there. Divide by 6.6 billion, and then you know how many cells there are. That's how many cells. It's not exactly how many neurons, but that's the basic idea. Okay, so now there's 84 billion neurons, and each one has a bunch of connections, and we want to study them. And that's what we're going to talk about. So this picture I have up here is not from the human brain. It's from a maggot. You guys like ever walking in the woods and you see like a dead deer with a bunch of like gross insects eating it? Yeah. Those are maggots. Anyway, people love studying maggots. They're also called larval Drosophila. Drosophila is a little fly, kind of like a fruit fly. And larva means it's still a baby, basically. So the mushroom body is part of the plasmid invertebrate brain. It's responsible for smell. It turns out smell is really important because animals use it to find the food and also to run away from scary stuff. Basically, like one thing that ants do a lot is if they smell a bunch of dead ants, they run the other way. It's a pretty good idea, right? You don't want to go where the dead ants are. So it's important for not getting smushed if you're an ant, and then it's also important for finding food if you're an ant. You smell and you're trying to find food. So smell is pretty important. So this part of the brain, the mushroom body, is responsible for learning the rules of smells. So what it does is it says, if someone trains it, or it happens to have been genetically encoded by evolution, that this smell is good and you should eat it, and this smell is bad and it will kill you, that information is essentially stored in this connection matrix, which I'll explain how to read what that is. Okay. You guys with me so far? Brains, they have networks. Here's an example network. This example network, there's 4 cell types, and there's lots of ways of thinking about cell types. This one has 4 of them. There's a Kenyan cell, which is whatever, it doesn't really matter what they are. With a Kenyan cell, there's a projection neuron, there's output neurons, and there's input neurons. But what this matrix shows is about 200 row column pairs. Each row column pair corresponds to 1 neuron. The rows are the inputs and the columns are the outputs. So if there's a green dot, what that means is there is a connection from one output to one input. Okay, and so what you can see is, for example, these input neurons get a lot of input from the Kenyan cells, hence the name input neurons. Okay? On the other hand, the output neurons, they send a lot of output up there to the Kenyan cells, which is why they're called output. Is this supposed to work out?
Yeah, I don't know.
Can you guys see that right now? Okay. Those are alpha guys. So there's the 4 different cell types, and what you can see, and I've just illustrated a little bit, there's a bunch of structure in this connection matrix, right? There's the out. These guys get input, and this and output. These guys get input. These guys get output and input, turns out. These Kenyan cells, there's some weird things going on. These ones up here, there's a lot of connections. These ones down here, there's not so many connections. There's really some cell types don't really have any connections with other types. It's just, there's lots of structure in this. And the idea behind connectomics and what we're going to talk about is that that structure is meaningful. And that structure encodes for you basically what to do. So imagine you're, say, a hyena, and you have a baby hyena. And you want that baby hyena to have certain behaviors that you have. And hyenas aren't that great at English or any other languages as far as I can tell. So how are you going to teach your baby hyena to do the things like run away from tigers, which you want your baby to do, unless you want them to be eaten by a tiger? How can you teach them that? You have any idea how you teach a baby hyena to run away from a tiger? Yeah, it's really hard. I don't know how to do that. I don't know anyone that do that. But nonetheless, hyenas have learned over the course of evolution to run away from tigers. So how do they know that? They're born with that knowledge. If you're a hyena, you see a tiger, you run away. I have no idea, by the way, if hyenas and tigers ever interact in the wild. They may live on different continents, though. So let's just assume for now that they live on the same continent and nearby, and that hyenas are very tasty for tigers. And you want your baby hyena to know to run away from tigers so it doesn't get eaten, so that you can have grand hyena babies. How does it do that? How does a hyena know to run away from tigers? Where is the information stored? Is it stored in its skin? Does its skin tell it? No. Okay, so sorry, not in the skin. Any other guesses where it may be or may not be? Is it in the fur? I just got a cool fur. Yeah? In the brain. That's a good, I'm into that. Where in the brain? How is the brain storing this information? Do you have a guess?
Yeah, like the connections in different areas of the brain are associated with different memories.
Yeah, I'm with you. So what she's saying is, you got a brain, and there's a whole bunch of connections, and essentially, you're a hyena, your baby hyena, and your eyeballs see a tiger. And that causes a bunch of neural activity from your retina down to a bunch of other parts of your brain that cause a cascade of activity through lots of different parts of your brain based on which parts are connected to which other parts that result in you turning around away from that tiger and running the other way. And the reason you do all of that is because the neurons from your eyeballs that information gets routed to different parts of your brain and eventually ends up in the part of your brain that controls your legs and makes you run in the other direction. So without your genome having encoded a particular statistical patterns of which parts of your brain should connect to which other parts, without the normal course of development where those connections are formed in the appropriate fashion, you won't learn to run away. You won't know how to run away from a cheetah, and you will be eaten by a cheetah, and then your children won't exist. On the other hand, all of those hyenas who actually did run away from the cheetah, or tiger, whatever it's eating it, all of those who ran away from it, whether it was totally random happenstance or not, the ones who had connectomes that tended to make the neurons in their brain turn around and run the other direction when they see a tiger, those animals, they had children that didn't get eaten, and the genes that they propagated down to their children encoded these probabilities. Right? So what I'm saying is, if you want your children to do cool stuff, you need to understand what connectomes are. That's basically it. Okay. But more seriously, your connectome is really a fundamental object of making sure you do the right kinds of behaviors in the right kinds of environments. And so if we want to understand how the brain works in general and how the brain stores information in particular, a natural place to start is by studying the connections in the brain. Okay? You guys all with me so far? All right. So now we all agree that connectomes are awesome, right? Does anyone still think they suck? If so, you should let me know. I can try to further motivate it. There's lots of other ways of motivating and caring about connectomes. So for example, does anyone care about like well-being? Okay, cool. There's one guy. So the World Health Organization estimates the global burden of disease for lots of different diseases. And it's kind of interesting what counts as a disease or not, but one thing that counts as a disease is depression. And according to them, depression is a leading burden of disease in the world. So of all diseases, cancer, heart disease, emphysema, diarrhea, various infectious things, whatever it is, depression is the most burdensome. And it's the most burdensome for a pretty straightforward reason, actually. Other diseases kill you. Right, so say you get something really terrible. What's a terrible disease that you might get?
Cancer.
What? Cholera, yeah, that sucks. Okay. Let's say you get cholera. It sucks for a while and then you're dead. Right? So that costs some amount of money. This is how they figure out burden of disease, how much money. If you're depressed, you don't die from it. Right? Depressed people can live for a very long time. But they also don't work. And so they cost society a lot of money by these calculations because they're around for 80 years and they're never contributing economically. Right? So it's like a double whammy. All the diseases where they kill people, it's expensive to treat them while they're alive, but then they die and it runs out. So depression is a really, really problematic economically for the world. And probably will continue to be more and more problematic as people figure out how to prevent other diseases than the relative impact of depression as compared to other things. So now why am I saying all that? It's widely believed within the psychiatry community that depression, learning disabilities, and other psychiatric disorders are all fundamentally connectopathies, meaning something about the wiring diagram of these individuals is different than other people in such a way that makes them experience the world in ways that are unpleasant. Okay, so if we want to understand learning disabilities or psychiatric issues or neurological degeneration, it is likely, or at least a lot of people believe, that if we understand how the connections are different in those kinds of people, then we'll be able to treat them or better yet prevent those kinds of problems. Okay, so that's why I care about this stuff. But just caring about it is one thing, and now having all this data that people have collected in whatever ways they collected it is another thing. And then there's a question of what to do with it. And so it turns out, for the last 10 years, people have been collecting these data and doing stuff with it. And for the most part, it's all nonsense. And that's okay. That's the typical thing in science. People start collecting data and you don't really know how to analyze it, so they do a lot of nonsense. But it doesn't have to be nonsense. There is a way, bless you, to make it not nonsense. And so what I'm going to spend the rest of the time talking about here is how to make the analysis of these kinds of data that we care about and we have be not nonsense or otherwise sense. And what I'm going to say is the way that we do that is with statistics. So you guys, you've all taken some stats test, right? You take 311 and or 420. Who took 420? Who took 311? Okay, so even 311, you learn. Okay, so I'll keep the math to 311 level basically. But in 311, you learned, I think, some basic fundamental things in statistics. And this is the way statistics has always gone the last 100 years since Fisher more or less made it up. The first thing that happens is you have an interesting question. All of statistics and all of science starts with an interesting question. The first statistics question ever, do you know what it was? It was pretty crazy and hilarious. Yeah?
Is it the lady tasting tea one?
It is the lady tasting tea. How did you know?
Carrie told me.
Oh, Carrie told you? That's cheating. So,
Who was like one of the first and
Yeah,
most
sure.
famous statisticians was at a dinner party. And the lady claimed there she can tell whether the tea or the milk was first added to the cup.
right.
And he like came up with an experiment that like giving her a randomized trial of
Right.
four of each, and like whether she was able to tell it more than a statistical chance.
Yeah, so you guys get that? This lady claimed in her tea she could tell whether you poured the tea in first or the milk in first, because you're going to pour both into this cup. And then Fisher basically called her bluff and said, that's bullshit, not true. And then he said, well, why don't we actually check whether that's bullshit? And he had to invent the concept of a randomized controlled trial. It wasn't quite a randomized controlled trial, but he invented the concept of a trial where he gave her, I forget exactly how many, let's say 10 examples. Half of them were tea first, the other half were milk first. He didn't tell her which one. Right? And then she guessed which one was first. And he calculated the probability that she was right as many times as she was, under the assumption that she didn't know. Right? And that was essentially the first p-value ever. I mean, it's kind of fun to like prove crazy ladies wrong, but it's not that important really. But it did start all of statistics, and it establishes, I think, for us, just this concept of the first thing we do is we ask an interesting question, and in the world of economics, it can be something like, how are brains of people who are depressed different in terms of connections from people who are not depressed? A very natural question. So that's where statistics always starts, and building scientific evidence always starts. The next thing that we do, always, is we write down a model, a statistical model. So you guys are familiar with lots of statistical models. Gaussian is one that I'm sure came up. Bernoulli, binomial, Poisson, exponential. These things are all familiar to you guys, right? Okay, so like for example, people say the distribution of heights in the US for males is Gaussian. Is that true? You guys think the distribution of heights for males in the US is Gaussian? It's not exactly true. For example, Gaussian says there's some positive probability of having a person whose height is -8. Right? Gaussian says. It also says there's a positive probability of your height being infinite. That's not right. Your height will never be -8 or infinity. No matter how tall or short you are, I promise you it is not negative or infinite. Okay? So Gaussian is wrong. It's okay. It's close. It's good. For lots of things, it's a really good approximation. And that is the job of statistical models, to be helpful. Their job is not to be correct. So as this will come up in lots of times for you as you're doing science, people write down models and they'll say that it is true. They're not true. You all know that because you're not having to do it. But they are useful. And so the next thing that people do is write down a useful model. And the reason it's useful is because you can do math and you can calculate things from it. You need to be able to write down the equations and calculate stuff if we're going to build quantitative evidence. The next thing that happens once they write down these known reducible models is they derive some estimates. So if the distribution of heights in the US were Gaussian, then you would be able to estimate the mean and the variance. It turns out you can estimate the mean and the variance even when the model is wrong, as we do. But you need a way of estimating the mean and the variance. And if it was Poisson, you could estimate the rate. If it was exponential, you could estimate the constant. And if it was this, you could. All of these different models, one property that they have is you can estimate the parameters of them. And then you can use that to test things or experiment. So you get these estimated. But the next thing that they had to do to really get strong evidence is to prove theorem. Right? So part of the reason that, like, who here believes smoking kills people? Okay, why do you believe it? What? Why do you believe it? Yep. There's carcinogen. That doesn't mean it kills people. There's lots of things. Everything's carcinogenic. Like if I just smash your face with glass a bunch of times, eventually you're going to get cancer in your face because your cells keep dividing. Every time your cells divide, it increases the probability there's a mutation. And cancer is a whole bunch of mutations. So everything is carcinogenic eventually. It just has carcinogenic. So, yeah.
It is
By the
known
way,
to
I'm not
carry.
smashing your face with that.
It is known to carry a large number of carcinogens, which increase the mutational burden of cells in the body, and statistically, with an increased mutational burden of cells, they are much more likely to become cancer cells. Which cancer is the second highest leading cause of death in the U.S.
Okay, great. So let me try to summarize what he was saying. What he's saying is, someone did an experiment and tried to check whether if you smoke more, do you get cancer more? And their answer was yes. And this is the mechanism you're proposing, this carcinogen thing. But whether or not there's a mechanism doesn't actually change that the data said people who smoke more get cancer more. Right? And sometimes you can do that. You can say, well, if the lady KCT got the answer right more than you expect by chance, maybe that's okay. But why is it in this case that when they conducted these experiments, like hypothetically, and they gave people a bunch of cigarettes and they died more than the people who didn't? Why can we generalize? Right? Why can we say, well, it's not just in those people that this happens, but that it's in these other, that would happen to us too? Why is it not just a description of what happened? Yeah.
Because he used like the idea of the representative sample where you basically like the average person and see what happens to them while controlling for everything else that happens to them should be like about the same,
Yeah.
like the one condition.
So I think what she's saying is there is some model of the population in the whole, and we believe that we've sampled well from it, and so that this is a representative sample. Right? And just because it's a representative, and I agree with all of that, and I'd say in addition to that, people have done that particular thing, gotten a representative sample, computed the test statistic in this way, found that the answer was, P was really, really small, and then concluded that this was a general principle, because the model said that this should be a general principle rather than just for this population. And they did that a whole bunch of times in a whole bunch of different settings, and it turns out it is often right when you do it that way. Right? So there's lots of empirical evidence supporting that if you do statistics this way, you get the answer right. And almost everything that has that property, that people have done it over and over and over again, that tends to be right, has a bunch of theoretical guarantees about when it's right. It's almost never been the case historically, scientifically, that people use the same thing lots of times, it turned out to be right, and there were no theoretical guarantees that said it was doing the right thing. So the way that we as a community, as a scientific community, build up trust in procedures is not just in writing them down and showing that works in some small numerical examples in real data simulation, but also having some kind of deep understanding of theoretical claims that it should work in this setting, and it shouldn't work in this setting. And so if we know the setting we're in, then we can say we believe the result. And if we don't know what setting it is, it's more complicated. So it's not just enough to write down a model and then be able to estimate stuff, but you really want to understand when it should work and when it shouldn't work, and that's what theory is. Theory just tells you when it works and when it doesn't work. There's nothing really more complicated about statistical theory than that. And the reason that's important is because nothing will always work. Everything will work in certain cases. So whenever there's a procedure, what you really want to understand is when it works. That's what theory does. And then, as we said, there are lots of experiments doing it. So these 5 things essentially why we rely on statistical evidence for any scientific claim. And so what I'm going to argue is that we need to do that also for connectomics. We want to make claims about when you're depressed, it turns out your brain tender looks like this rather than like this. If we're going to believe those claims, we want to have foundational statistics justify that we know that we're in the assumptions the data look like, it's a good model for the data, such that we know when the answer is the p-value is really small that we should trust it. And if we don't have that, we really don't know whether we should trust it. And there's a lot of science what people do, they just kind of write down stuff and you don't know whether you should trust it or not, and it turns out not. But if you do it this way, then you feel more confident. Is that clear? Does anyone have any questions about any of that? Because that was like motivating all of statistical theory in like a slide, so it's kind of a lot. Does anyone have any questions about any of that? Okay, so the first sentence on the slide is basically a bunch of old dead dudes and women did all these things. And we want to do it now in connectomics, but those dudes are dead. So I found this guy. He's not dead yet, he's very old. He's like 100. But, so he and I did a lot of this work together. Okay, so, what kinds of questions are we going to try to answer? I'm trying to motivate things with the psychiatry and with memories, but the first study ever in connectomics was done by this old, now dead dude. He actually just died like 3 weeks ago, Sidney Gregg. He did it on a C. elegans. It's a little worm, has 302 neurons. Actually, there's a male and a hermaphrodite. The male has 302 neurons, the hermaphrodite is his number. And what he says, and he won the Nobel Prize for this work, and he says in his Nobel Prize lecture that neuroscience is fundamentally after answering 2 kinds of questions. One is the genetic specification of the nervous system. And that's what we talked about before, right? If you're the hyena, you give a genome to your children that specifies how the nervous system is developed such that it runs away from the egg. And the other is how do nervous systems work to produce behavior? So that is, now you have your connectome, and what are the patterns of activity that how they propagate through the connectome to generate your behaviors. These are just 2 fundamental questions about how the brain works. Both of them require understanding the structure of the brain, what's connected to what, and also other stuff. And so I'm going to formalize this thing that he's saying as just writing down 2 different conditional probability distributions. So everyone here knows what conditional probability distributions are, right? Okay. So one is, what is the probability of connectome given a genome? Right? So what we would like to be able to do is, you say, here's my genome, because you went to 23andMe or whatever, and they sequence your whole genome. I don't know if they actually do that, but sequence on it. You give it to me, I know all 6.6 billion base pairs, and I say, okay, this is what your connectome is going to look like, with some probability. And with some other probability, it looks like this, another probability, it looks like this. I would like to be able to say, given any one genome, I can tell you the probability that your connectome will look like anything. Okay, that's this first thing, the genetic specification of the genome. The second thing is now, you tell me what your connectome is, and then you put me in any context, and I tell you what's the probability that you'll behave in any particular way. So let's say you give me a hyena connectome, and I put a tiger, say there's a tiger there, I should tell you what kind of behaviors it'll do, just based on knowing its connectome and the surrounding environment. That's the other question he wanted to know, which is how the nervous systems work to produce behavior. So he can say it like this, but we can formalize it as statistics questions. We want to estimate these 2 conditional probabilities. Okay? Does anyone have questions about what these two conditional probabilities are? Okay, so they're, both of them, as you'll note, have this word connectome. Nope, not that connectome, connectome. Both of them have, they're assuming connectome is in there and it's a random variable. And so if we're going to do either of those things, seems like maybe a good first thing to do is just write down probability of connectome. In other words, write down statistical models that might characterize our connectome. And that brings us to this, which is the connectome that we're going to build a statistical model for. And I have human connectomes and sea lion connectomes and coyote connectomes and primate connectomes. There's lots of them, but this one I like because it has a pretty cool structure. But everything I'm going to say about the mathematics and how to do this, you can apply to any connectome. Okay, so let's say we're going to build a statistical model for this connectome. Now, I don't want a statistical model just for the edge. I want to be able to, what that means is I want to be able to sample the connectome. Right, so now I'm going to ask you guys to tell me how you might sample this entire connectome. Right, not like a feature of the connectome, but give me a procedure for sampling the whole thing. Let's do the simplest, see if you can think of the simplest possible way of doing it. All of these things are binary. Right? Each dot is either on or off, and there's about 200 squared of them. So what's the simplest way to sample something like this? Let's just say there's only one potential edge. What would you do there? It's like you have a coin that can land on heads or tails. You flip it. Throw it up in the air, it lands on heads or tails. Now you've sampled a coin that either landed on heads or tails. Okay, so now you know what I'm talking about. Now let's say you have 2 coins. You could land on heads or tails. So what are you going to do? What's a really simple thing to do? Yes. Yeah.
Flip one and then flip the other.
That's a good idea. Flip one coin and then flip the other coin. Now we need to specify an actual probability distribution rather than a model. So what's the probability that the first coin lands on heads? Yeah, you're the one coming up with this generative model. 50%? Okay, what's the probability that the second one lands on heads?
50%.
Are they correlated at all? No, okay. That's a perfectly fine model. And in fact, Erdos and Rainey made up that model and named it the Erdos and Rainey model rather than whatever your name is model because they said it first. But if you were born before them, then I would have named that for you. And the basic idea is all of these things, there's 200 square potential dots. Every single one, they just flipped a coin independently with some probability of landing on heads. And it's the exact same probability for each one. So they start with this dot, they flip the coin, it's heads, they get the dot. And the next one is tails, there's no dot. And then the next one is heads. And they did that 200 square times, and then they got this one. So the key to the Erdos-Rainey random graph model, which is the world's simplest possible random graph model, is that every single edge is independent and identically distributed. I mean, just flip coins totally separately, and each coin has the same probability of landing on it. Okay. Yes?
What's an edge in this context?
An edge is a dot. Sorry. Is whether or not there's a connection between the pair of neurons here. Sorry. Good question. Any other questions? Okay. So that's the simplest possible model. How many parameters are in this model?
One.
One. Yeah, there's one, right? It's the probability that any of them are going to land on heads or have a dot would be an edge. Okay. Do you think this model is going to do a good job differentiating between, say, people who are experiencing depression versus people who are experiencing, say psychopaths? I think if we fit, we can measure the connectomes. We're going to fit all the people that say we're going to take a bunch of psychopaths, measure their connectome, estimate this model, and that's going to have one parameter, which is basically the probability there's a connection between any pair of regions in their brain. And we're doing it for a whole bunch of depressed people. What do you think is going to differentiate between the two populations? Probably not, right? I mean, maybe. But there's lots of cases for which it's very unlikely that that is going to be a sufficiently rich and complex model. So let's make it more general. So how are we going to generalize this model? What's classic?
Yeah.
Okay, I'll tell you that later.